Welcome, everyone. Thank you for tuning into this masterclass on how a dysbiotic microbiome becomes toxic to the brain and the emerging roles of psychobiotics. I'm your host, Morgan Justo. I am the Research and Development Manager at Microbiome Labs. As most of you know, I have Kiran with me today. Kiran is the Chief Science Officer at Microbiome Labs. Kiran is a research microbiologist. He has been part of the dietary supplement industry for the past 19 years. He is a hands-on researcher and developer in the fields of molecular medicine and microbiology. He is a leader in the microbiome and probiotic research and has also been part of many clinical trials regarding human nutrition. Kiran is going to talk to us today about how a dysbiotic microbiome can become toxic to the brain through various pathways and the roles of psychobiotics during about a 40-minute presentation. After that point, we are going to have a Q&A. So feel free to ask questions in the Q&A box on the bottom right panel, and we will try and answer as many questions as we can. In the upcoming days, you will also receive a recording of this webinar to your registration email. Thank you so much, Kiran, for being with us today.
It is my pleasure. Can you hear me? Okay.
Yeah.
Okay, sounds good. Thank you, Morgan. Let's see what happened to this little thing that disappeared up here. So I think we're still waiting on some people to come in, right? So maybe we'll give it another minute or so, and I can give people a little bit of background on this particular topic and our role in this whole world of psychobiotics, which is a relatively small world at the moment, because I think this is one of the first official commercially available psychobiotics on the marketplace, at least one with a significant amount of research behind it, showing the mechanism of action of how it functions. We got to partner recently with Novozymes One Health. Novozymes is a large Danish biotech company with a huge focus now and investment into human health and human nutrition. Novozymes is an expert in enzymes. That's how the company was founded a number of years ago. Novozymes has something around like 13 million enzymes. They're the largest enzyme producer in the world, but they also have a massive focus on the microbiome and probiotics and all the good stuff that comes along with that. So they have around 36,000 probiotic strains in the library and a very, very active R&D, really massive team, which includes some of our MBL folks as well, and including myself, in terms of developing and screening for new functional microbes. One of the really hot areas is in partnership with a company called Psychobiotics out of Ireland. And that hot area is this very area of the gut-brain axis, right? Because the gut-brain axis becomes important in a number of different ways. Number one, it's important when it comes to neurodegeneration. When we're talking about degeneration of the central nervous system, the brain, age-related degeneration, which leads to conditions like Alzheimer's, dementia, Parkinson's, and so on. There's also the cognitive functional aspect of the brain that becomes really interesting and exciting for people. So we're talking about the advent of nootropics and other things that can enhance the functionality of the brain. And then, of course, the third part and presumably one of the largest areas is the mood component of the brain. So looking at things like stress management, sleep, anxiety, depression, and so on. So there's many components of cognitive function and brain function, neurological function that are really becoming extremely relevant right now and need some sort of innovation, because many of these areas have not seen innovation in decades, right? So if you look at the typical practice of psychiatric medicine, the options there from a prescription standpoint have not really changed much in the last several decades. It's the same kind of SSRIs and anti-anxiety meds in kind of different flavors, but the approach hasn't changed at all. And what the microbiome gives us is a very, very unique opportunity in truly understanding the pathologies of what impacts the brain and how that impact leads to neurodegeneration in the case of age-related dysfunction or cognitive dysfunction, slowing down of the memory, losing short-term memory, slower cognition, lower IQ. And then, of course, the final part, which is what we'll talk about today, that's the mood and sleep component of brain function as well. So we're heavily involved through our partnerships and working with Novozymes and Precision Biotics on bringing a whole bunch of solutions, hopefully in the next few years. This one in particular is one of the most exciting that we've been able to do, maybe since Megaspore itself. So very excited to present this. Should I go ahead and jump in, Morgan?
Mm-hmm. Yeah, you're all good to go.
Any housekeeping notes on the whole area of questions? How do you want them to ask questions? Do you want them put in chat box or in the question and answer session?
So we will do the question and answer in the Q&A button down below, not the chat box, but it's the Q&A button. So we will do it through that.
Perfect. Okay, so I'll go ahead and I'll turn my camera off so I don't have my little bubble of my own head bopping around when I'm talking, and I'll jump right into it. So as I mentioned before, we're heavily involved right now in gut-brain exploration in a number of different frontiers. This particular topic, with what we're going to show you today in terms of psychobiotics, really has evolved over the last about 8 or 9 years. So this is a combination of a lot of research work by some of the top researchers in this field through the APC in Cork, Ireland. The APC is one of the preeminent research institutes for gut-brain research and innovating in that space. So we're very lucky to be able to work with these groups and be able to bring these solutions to you, to the US market itself. Now, as we start looking at the gut-brain axis, one thing that's really clear is there's a very intimate connection between the gut and the brain, right? To start off, when you look at embryonic tissue itself, the gut and the brain basically evolved from the same components of embryonic tissue. As you guys know, embryonic tissues can differentiate into all the different tissues that make up the human body. But what's interesting is you look at the development of the brain and the gut lining in particular, they both develop and differentiate from the same parts of the embryonic, the embryo itself. So they're intimately connected from day one. And then, of course, when you become a full-fledged human, they become very intimately connected through the vagus nerve. And that's connecting the enteric nervous system in the digestive tract all the way up to the brain. And it is bidirectional. Now, that's not the only way that the gut and brain are connected. The gut and brain affect one another almost equally in many respects by a number of other mechanisms. One is through the enteroendocrine system, which we have a number of enteroendocrine cells that are embedded among our intestinal epithelium. These cells produce endocrine molecules, and these endocrine relevant molecules are being produced in the gut lining by influence of microbes, influence of your diet, influence of your environment, and these can make their way to the brain directly through the vagus nerve, and in some cases through circulation as well. We also have heavy involvement of the immune system. That's one of the things I really want to touch on today. Is it's not often talked about how much the immune system is involved in activation in the brain and response, both cognitive response, physiological response systemically to in terms of the host, with the influence of the immune system, right? So the immune system plays a central role in signaling between the gut and the brain, and also in response to external stressors and deviations in the microbiome in the gut as well. So that immune response within the body, within the gut, which then translates to the brain, is really quite a potent response and is an important checkpoint in how we respond to stress and how we respond to the world around us. Then, of course, there's a number of postbiotics that the microbes make, things like GABA, 5-HT, BDNF, which is brain-derived neurotropic factor. Of course, serotonin, dopamine are also made in the gut. All of these things can be made in the gut by microbes or stimulated by microbes. For example, serotonin is stimulated by certain microbes, but it's made by the enterochromaffin cells. But our microbes stimulate the enterochromaffin cells to produce the serotonin. Those, of course, can also make it to the brain through the circulation, through the lymphatic system, or even through the vagus nerve itself. So there's intimate connection. It's bidirectional. The gut does influence the brain, and the brain does influence the gut as well. And of course, that deep connection through the vagus nerve goes through the enteric nervous system. And the enteric nervous system, the most important thing to note is it, of course, covers your full digestive tract. Your microbes that live in the digestive tract have full access to it. And it is a highly dense neurological tissue. So there are over 100 million neurons, more than we have in our entire spinal cord, that are found in the enteric nervous system. So the second most complex neurological organ in your body is actually on your gut, and your microbes have full access to that. The other thing that's important to note is almost every known neurotransmitter can be found in the enteric nervous system. All of these, inhibitory or excitatory transmitters, all of these that have been discovered and known to function in the brain, known to circulate through the endocrine system in the body, are also found in the enteric nervous system, many of which are also made in the gut, or if not, metabolized in the gut. So many of these cycle through the gut as part of the normal feedback loop and feedback inhibition that is supposed to occur for these types of neurotransmitters. We'll focus on one neurotransmitter in particular, cortisol, and we'll talk about the impact of glucocorticoid dysfunction in terms of negative feedback and how the gut plays a role in that. But other hormones that people know well, a lot of information about in terms of the gut's role in cycling is like estrogen, for example, right? So estrogen dominance is a factor of having less estrobolome, fewer microbes that metabolize estrogen properly in the gut as estrogen gets dumped into the gut. So you get more resorption of estrogen and you end up with a higher level of circulating estrogen. And that checkpoint is in the gut. The gut microbes, the right type of gut microbes, are supposed to be metabolizing it. So many of these neurotransmitters have checkpoints and negative feedback or metabolization pathways in the gut to regulate their levels and regulate the function. Now, I mentioned it's bidirectional influence between the gut and the brain. And of course, as a host, we have ways of impacting our gut microbiome through behavior. Of course, we know diet and exercise and other lifestyle factors are a key thing as well. But one of the things I really want to point out and mention here is the stress and mood component of it. And we'll talk about stress a lot, but as we allow ourselves to experience heightened levels of stress or extended periods of stress, one of the effects of that stress is the release of glucocorticoids like norepinephrine, cortisol, and so on. All of these have an impact on the gut microbiome. And in particular, norepinephrine and epinephrine can actually increase virulence factors of opportunistic organisms within the gut microbiome or within the systemic microbiome as well. So, for example, one of the reasons why these are opportunistic organisms, the ones I'm focusing on, is because they look for the right opportunity to express their virulence factors, right? They're not direct, strong pathogens. So they know that if they express their virulence factors readily all the time, that the immune system would easily detect them and get them under control. So they look for the right opportunity, and they've evolved to learn that one of the best opportunities to express their virulence factors is when the host is under stress, because when the host is under stress, the immune system is suppressed. And so they've picked up this evolutionary capability of reading epinephrine and norepinephrine and cortisol levels in the system. When they detect high levels of those glucocorticoids, they will actually start expressing the virulence factors and start to grow more readily. So even short bouts of stress over a period of time can start to change what your microbiome looks like and actually increase the level of pathogens within your system. And that's a really important thing to note. And we'll talk about more of that implication through the gut itself later on. But just keep that in mind that we will impact our own microbes through our perception of stress and the way we manage stress itself. So it becomes a nasty, self-perpetuating problem. And again, I'll clarify that a little bit more when we talk about this later. When you look at neurological disorders with associated gut dysbiosis, there's been lots and lots of studies in animal models, and then some good studies on human systems as well. So a number of things have been verified in humans. Now, when you look at in the mouse model, all of these types of conditions, things like autism spectrum disorders, schizophrenia, Parkinson's, Alzheimer's, depression, MS, Huntington's, anorexia, have all been demonstrated to have an association with a certain type of dysbiosis, right? And those same things have also been shown to be true in the human system. And then there's a lot of other conditions that have been studied further in mice with very specific changes in microbial population within the gut that drive these conditions, right? So things like antibiotic impact, how the use of antibiotics can actually, create reduce the risk of multiple sclerosis, Parkinson's, Alzheimer's, if it's a pathogen-driven condition. So what they do is they'll inoculate these mice with high levels of pathogens, so mimicking a dysbiosis, and then they will actually raise the mice that will start to develop these types of symptoms, these cognitive dysfunction symptoms. And then when they use an antibiotic to kill off those pathogens, they see a reversion of some of those symptomologies. And they've done similar studies to this on autism spectrum disorders, for example, in mice, where if you inoculate mice with the right type of pathogens like Clostridium bolte, it increases spectrum-like behavior in these mice. And then if they can kill those pathogens using an antibiotic, those conditions kind of go away. The symptomologies go away. Of course, from a human perspective, this is not a tenable solution because you can't be taking antibiotics forever. But what that demonstrates is a direct effect of reducing certain types of organisms and then changing symptomology, right? Or even enhancing the growth of certain organisms, like in the studies that they've done in probiotics, where you enhance certain types of organisms and things like Alzheimer's, anxiety, autism spectrum behavior starts to go down. So there's been a lot of this kind of work where you can tweak and manipulate the microbiome and see direct effects on outcomes. All of this has been done in mouse models, but then in human models, there's been really, really good, strong mapping studies that have been done to show that there's certain types of microbes within the human system that drive conditions as well in symptomology. One of the things that's really interesting to note is we also all have significant influence on one another and one another's microbiome, right? So it really does kind of take a village to raise a healthy village, if you will, because in any given household, all of the inhabitants of those households share microbes through the environment, through contact, and so on. So this study is really interesting. We call this like a microbiome cloud that surrounds you and your most frequented ecosystems, right? Your workplace or your home where you live. This was a study that was interesting where these researchers took individuals that were prescribed an antibiotic for whatever condition it may have been. And then prior to these individuals starting the antibiotic, they took microbiome samples, and then they took microbiome samples during and after the antibiotic treatment. And they went as far as 6 months after the antibiotic treatment. And as you would expect, they saw a significant dysfunction or dysbiosis within the microbiome once these individuals started taking the antibiotic, and then throughout the course of the antibiotic, and then of course upwards of 6 months after they were done with the antibiotics. But what was surprising about this study is they also followed the gut microbiomes of individuals who are household contacts with the people taking the antibiotics. And mind you, the household contacts were not on antibiotics. And what they found was that the household contacts also saw similar dysfunction in the microbiome as the person that was taking the antibiotics themselves, right? So there's a clear line here where they're demonstrating that any one individual with a significant disruption to their microbiome, in this case, using a course of antibiotics, can really disrupt the microbiome of a lot of the other people around them, especially within the same household. And so we have a negative influence on one another. If one person in your household is not healthy, is not making the right choices, has a dysfunctional gut, it can perpetuate dysfunction among other people within the household. So it becomes really important to really understand the role of the microbiome in this, because it's not that individual's behavior is going to influence the household inhabitants in the same way as their microbiome can, in fact, influence in a negative way the household context of that individual. So their behavior may create quality of life issues and so on, but their microbiome can actually set up their household context microbiomes to become more of a depressive or anxious microbiome. Now, when we look at the pathologies and the mechanisms that really impact the brain, especially when we're talking about mood dysfunctions, neuronal degenerations, and so on, there are really two big offenders, two big pathways that we have to pay attention to in order to really understand the problem and understand the root cause of the problem. And the two big offenders are LPS endotoxemia, so the presence of LPS endotoxins in circulation, and those endotoxins making their way into the central nervous system. And then the second is stress-induced HPA activation, right? So these are two different pathways, all end up with inflammatory type of activation, but they affect the brain in different ways. And we'll go through that in a moment. But these are the two biggest offenders of the things that are harming your brain that impact cognition, mood, longevity, and so on, right? So let's take one of these at a time. Let's start with the LPS, endotoxin-driven inflammation. We've done a lot of work on LPS ourselves. And as you know, if you've ever seen any one of our talks, there's lots of research on the physiological impact of having high levels of LPS in your system. Everything, of course, within metabolic syndrome and autoimmune conditions, all of these things are driven heavily by LPS, by having elevated LPS levels. But LPS is extremely pervasive. It enters every part of the body. And as it turns out, it is really profoundly able to impact the brain. So here's some of the studies that show that when you start to look at how leakiness in the gut can really impact the brain, you start to understand that LPS plays that significant role. So when our gut lining is healthy, we've got high diversity, we've got protective strains, we've got high production of short chain fatty acids and well-formed tight junctions. You've got this real distinct layer, this mucin 1 and mucin 2 layer. This mucin inner layer, this mucin 2 is thicker, it's relatively sterile. You don't have a lot of microbes living in that area. And this mucin 2 layer becomes an important communication conduit between the intestinal epithelium, which also contains lots of immune cells, and of course the enteroendocrine cells as well, and the crosstalk that these cells do with the microbiome that's out here. And this layer up here would be more like the lumen. That's where the lumen would be. Below the intestinal epithelium would be where the circulation is. So this inner part of the mucosa is so important. That's where most of the sampling occurs. That's where the communication occurs between the microbiome, our immune system, and our endocrine system as well. And it requires high diversity, high levels of protective strains like keystone species, high production of short chain fatty acids, and well-formed tight junctions to maintain the homeostasis of this area. Now, what tends to happen with the process of dysbiosis is you start to get a change in the microbial ecosystem in the top part of the mucin layer, right? So let's say you're getting exposed to antibiotics or poor dietary choices or bad food choices or too much stress, for example, will start increasing the growth of opportunistic and other dysfunctional bacteria. And many of these bacteria tend to survive by eating away at this mucin layer. And as they eat away at the mucin layer, what they drive is a translocation of not only bad bacteria, but also many of your own commensal good bacteria into the inner part of that mucus layer. And with that, you start getting dysfunctions and breaking up of the tight junctions. You get a lot of immune recruitment to the intestinal epithelium. That immune recruitment and that inflammatory response damages the intestinal lining. That damage to the lining will then allow things like LPS to leak through every single day. And once LPS is in circulation, it can enter the brain quite easily. And in the brain, it'll drive all kinds of inflammatory damage, right? So that's just how simple dysbiosis can lead to brain inflammation and eventually brain damage and other issues that are driven by that type of neuronal damage in the brain. So it's that simple dysbiosis, that simple imbalance of organisms where you get too many organisms that are eating away at that mucin layer and not enough organisms that are rebuilding the mucin layer. Then you get a net reduction in mucin, a massive translocation of all of those microbes and viruses and food particles and environmental toxins and all that to the inner part of the mucosa. That causes the intestinal epithelial cells to get alarmed and start recruiting lots of innate immune actors to that region. And when they recruit innate immune actors, there's going to be inflammation, there's going to be tissue damage. Your intestinal epithelial cells will get damaged at tight junctions, fall apart and stuff starts leaking through. And in particular, when LPS leaks through, it's going to wreak havoc on the brain. So here's some of the examples of that. This is a animal study. So I'll show this first because of some of the known mechanisms here. What they were looking at is in male and female mice, the effect of gut-derived endotoxins, in particular LPS, and its ability to impact repetitive behaviors or what they call sickness responses, right? So they concluded in here that in spite of the ubiquitous nature of LPS in the gut lumen, because remember, LPS is made in the lumen by gram-negative bacteria, and it's present in the lumen all the time. But if it's allowed to leave the lumen, leak through the lining and enter circulation, that's when it becomes a big problem. So what they showed is that this is the first study to demonstrate that intestinally derived LPS can initiate behavioral aspects of the sickness response. And they showed that while an increased enteric load of LPS increases anxiety, like behaviors in both sexes, it does so by a sex mechanism, because they showed that in male mice, the effect was even more profound than in female mice. So there may be some protective mechanism of estrogen metabolism here that helps protect the gut of the female mice a little bit more than the male mice, but it does increase anxiety-like behavior. And then the same kinds of studies have been done on humans and followed through. This is a study that was, I believe, published in 2020, so it's a most recent study. And they looked at basal and LPS-stimulated inflammatory markers in the course of individual symptoms of depression. The whole hypothesis here was that inflammation is going to be predictive of the severity and course of a subset of major depressive disorder symptoms, especially the things that we call sickness behaviors, right? And these sickness behaviors are really a critical part of the utter dysfunction that major depressive disorders seem to put on an individual, because these sickness behaviors are things like anhedonia, which is the inability to feel pleasure from anything, right? So nothing motivates you, nothing makes you feel happy, nothing really energizes you. So it becomes really hard to live any sort of good quality of life. Anorexia, low concentration, low energy, loss of libido, psychomotor slowness, irritability, and malaise. These are the symptoms that they were looking at. And what they found was that basal and LPS stimulated inflammatory markers were more strongly associated with sickness behavior symptoms. So they're looking at individuals that had elevated basal and LPS stimulated inflammation compared to those that did not. Those that did had more association with these types of sickness behaviors, and they followed all of these individuals for up to 9 years to see if that was still true. So it wasn't just some temporary fluctuation in those individuals' behaviors. And they found that even up to 9 years follow-up, people who had elevated LPS, elevated basal inflammation in their system did have higher levels of those sickness behaviors. And then when you look at other studies have looked at cytokine production in depression and anxiety. This is a big study that came out of Netherlands. It's a Netherlands study on depression and anxiety. This is over, I think, 1,200 patients. And what they found is that LPS stimulated inflammation was associated with increased odds of current depression and anxiety disorder. So the more LPS-based inflammation you had in your system, the higher your odds were of having anxiety and depressive disorders. They also found that LPS-stimulated inflammation was associated with severity in depressive and anxiety symptoms, right? So not only is having LPS-induced inflammation in your body going to drive your risk for developing depression and anxiety, but the level of LPS-induced inflammation will dictate the severity of the symptoms as well. And they talk about, unlike basal inflammation, so that's your normal chronic inflammation, typically through the arachidonic acid pathway, LPS-stimulated infection was still associated with anxiety symptoms and severity after adjustment for lifestyle and other health factors. So taking out people with certain chronic illnesses and so on, when you looked at them comparatively, LPS-based inflammation was always a marker for severity and risk for anxiety and depression-like behaviors. And in particular, markers like interleukin-6 interleukin 8, MCP1. These are all really critical markers, TNF beta. These are the key interleukins and markers of LPS associated or LPS stimulated inflammation in these patients. You also have microbiome-derived lipopolysaccharides that have been found to be a major driver of neurodegenerations in the case of Alzheimer's. So this paper in 2017 found that they were able to enrich LPS, microbiome-derived LPS, in the perinuclear region of the brain of people suffering from Alzheimer's. Prior to this, it was known that there's some sort of innate immune response going on in the brain that leads to the neurodegeneration, the dying off of the neurons, the beta plaquing, and so on. But what was unclear was what was driving that innate immune response. And this paper showed definitively that it was lipopolysaccharide, bacterial lipopolysaccharide from the gut, leaking through the gut lining, making its way up to the brain and initiating that type of inflammatory response. This same thing, gut permeability and the presence of endotoxins and the inflammation associated with the presence of those endotoxins also drives the pathology of Parkinson's, right? And that's from the accumulation of something called alpha-synuclein. So alpha-synuclein is a compound that aggregates when you have lots of neuroinflammation from the presence of endotoxins. And that starts in the enteric nervous system. You can actually start to see the accumulation of alpha-synuclein in the enteric neurons, and then it moves up the vagus nerve and eventually gets into the central nervous system and then starts affecting motor function, which is the classical symptoms of Parkinson's. But the early symptoms of Parkinson's, the well-known early symptoms of Parkinson's is constipation. As the alpha-synuclein aggregates in the enteric nervous system, it's disrupting how the enteric nervous system transfers signals from the brain to the gut for peristaltic activity and for bowel movement. So the gut becomes constipated. And that's, again, all being driven by inflammation from the presence of LPS in the enteric nervous system. And as that inflammation moves up the vagus nerve, it also stops at the olfactory nerve, where it starts inhibiting the ability to smell or at least distinguish smells from one another. So that inflammation is another early onset symptoms of Parkinson's. And then from the olfactory nerve, it moves up into the central nervous system. Then it really starts affecting the normal, typical symptomology of Parkinson's. So again, gut permeability, driving translocation of endotoxins, driving neurological inflammation in Parkinson's, in Alzheimer's, in major depressive disorders, in anxiety, and everything. LPS is playing a role in all of this. We also know that LPS plays a major role in metabolic conditions. In this case, LPS has a capability of entering into the hypothalamic region of the brain and expressing low-grade inflammation in that part of the brain, which then disrupts the hypothalamus from being able to read your insulin levels or your sugar levels in your blood, right? So this drives something called central insulin resistance, where it's irrespective of body weight. So it doesn't matter. You don't have to be obese to suffer from this. Your pancreas could be working just fine, producing all the insulin you might need, but your brain cannot read your blood sugar levels. So it cannot regulate the expression and the release of insulin and glucagon. Right. So this is happening to people at younger ages of normal body weight, and it's all being driven by intestinal permeability and the elevated presence of LPS in circulation. And so this is one of the most toxigenic effects on the brain, right? We've been talking about things that are impacting the brain's ability to read critical signals within the body. For example, the blood sugar levels. We've been talking about how it can drive neurodegeneration in the case of Alzheimer's and Parkinson's, and also how it can drive not only the risk for anxiety and depression, but the severity of the anxiety and depression, and then in particular, the presence of sickness behaviors. And of course, there is this clear pathophysiology, as the study showed, that the gut-brain barrier in major depressive disorders shows clearly that intestinal mucosal dysfunction with that increased translocation of LPS, which of course comes from gram-negative bacteria, in this case, they focus on enterobacteria, plays a role in that inflammatory pathophysiology of depression. So that is all starting in the gut. And they also in this study talk about chronic fatigue syndrome. They talk about the pathophysiology of that being almost identical to that of depression, right? So it's just different parts of the body that this same translocation of LPS is affecting. So one of the big goals in order to protect the brain and support healthy cognitive function, support the right type of neuronal development and regeneration and all that, is to really affect the barrier and stop LPS translocation. If we can stop LPS translocation, we are taking care of a big chunk of the problem. It's not the entire problem with respect to the brain, but we are already taking care of a big chunk of the problem. So, of course, the best way to do that, as we've demonstrated through our published studies, is Megaspore. Megaspore is, as far as I know, one of the only maybe the only probiotic that has been shown to improve postprandial or metabolic endotoxemia and significantly reduce LPS levels. And in fact, in our published study, we showed over a 60% reduction compared to the placebo group in just a 30-day period in the amount of LPS that was present in circulation of these individuals with severe leaky gut. And keep in mind, these individuals were quote unquote healthy normal, meaning they didn't have any major chronic diseases or weren't on medications and so on. And yet they had severe intestinal permeability and lots of high levels of LPS postprandially. And we were able to shut that down quite dramatically in just 30-day period. Now, that makes a big difference. But what's also exciting is when you look at a heat map analysis of all of the inflammatory markers between the placebo group and the spore treatment group, you see a significant reduction in inflammation in the treated cohort versus the placebo cohort, which actually saw an increase in inflammation, which of course then goes along well with the fact that they saw an increase in endotoxemia as well. So if you're not familiar with the heat map, here, this is this column here is the spore-based individuals comparing their baseline to 30 days after taking the spore probiotic. And then this one is the placebo group comparing their baseline to 30 days after taking the placebo. And in the case of a heat map, the more hot the colors become, meaning more red and orange, the worse the deviation is from the baseline in a negative way. And so that is what you get to see the picture of with all of these important variables. And I, in particular, highlighted or circled interleukin-1 beta, interleukin-6, of course, interleukin-8, MCP1. Remember, those are all the markers that were talked about in those other papers that are the markers of LPS-induced inflammation, the same LPS-induced inflammatory markers that are also associated with higher severity in those conditions. In that case, they were talking about anxiety, depression, and so on. So being able to affect the microbiome in a positive way, reduce that endotoxemia, also brings down those key markers in circulation. And in fact, in the placebo group, all of these markers went up in that 30-day period, right? So Megaspore is already doing a huge service to your patients and your clients by reducing or arresting that LPS translocation. And we saw this effect in a gut-brain example in that same study, which is the effect on ghrelin. Ghrelin is a hunger hormone, right? So ghrelin is being released by the brain in response to what's happening in the gut. So if the gut is empty and the patient is fasting, you're going to start seeing an increased level of ghrelin being released. Now, once the individual eats food, there's supposed to be a feedback loop from the gut back to the brain saying, stop producing ghrelin. In fact, produce leptin, which is a satiety hormone that also starts stoking the metabolic process, right? So when you end up with leptin or ghrelin resistance, that is one of the fundamental aspects of obesity and weight gain issues. And so what we saw at the baseline of the spore treatment group, this is at the day 0 before they started taking the spores, is that when they would come in fasted, which is this blue bar here at time 0, their ghrelin levels would be elevated, which is normal because they're fasted, right? 12 plus hours. But we'd give them about a 2,000 calorie meal at that point, and their ghrelin levels over the next 3 hours would barely drop. And then it would actually start to increase again right after that 3-hour mark, right? So they're not getting the proper signaling on satiety, on kicking up the metabolism and stopping the production of the hunger hormone, because there's no reason why you should have more hunger hormone surging just 3-4 hours after eating a meal, right? That's not the time to go back out and get another meal. And so when we gave them and treated them with the probiotic for 30 days, and then changed the microbiome and reduced that endotoxemia, reduce, of course, all of those inflammatory markers that we showed, then you see a reestablishment of the ghrelin function, where their baseline ghrelin levels were already lower to begin with. But then once they were given a meal at time zero, you see this dramatic drop in ghrelin over the next 3 to 5 hours. And that corresponds to an increase in leptin as well. So this is a perfect example of reestablishing better communication between the gut and the brain. And it was so great that we saw this. We didn't expect this, but we saw it because it demonstrated to us that when these individuals' guts were leaky, that their gut and brain aren't talking properly, right? And if we can reduce that leakiness in the gut and brain communication reestablishes itself, right? So this was so exciting to see, and we've done other metabolic studies using the same principle, which we hope to publish sometime in the next 6 to 8 months. So LPS is clearly, without a doubt, right? There's ample research and really, really strong understanding on the mechanisms of action on how LPS from the lumen translocating through a leaky gut, ending up in circulation, is extremely toxic to the brain in many respects, right? We're talking about in the mood disorder issues, in the neurodegenerative issues, in the aging related issues. And we also know it can completely disrupt gut-brain communication, which can have all kinds of other profound effects. But the other part that we have to address is the stress-induced HPA activation, right? So that's what I'll talk about now. External stressors, so things like interpersonal conflicts, social isolation, and so on, drive a really interesting pathophysiology in the body, right? So external stressors are perceived by the HPA axis, so in particular by the hypothalamus and the pituitary gland. And once that is perceived, once an external stressor is perceived, that causes the nervous system stress circuitry to get turned on. And one of those first steps of getting turned on is the production and release of corticotropin releasing hormone. That once the corticotropin releasing hormone is released, that then stimulates the release of ACTH, and that ACTH is going to excite the adrenal glands to produce more cortisol, right? So once cortisol is released, that starts to turn on some of these stress responses in the body. Now, at the same time, the release of corticotropin releasing hormone from the hypothalamus will also start to turn on the sympathetic nervous system. And the sympathetic nervous system is going to have all kinds of output that impacts your physiological response. So it does it through alpha and beta adrenergic receptors, and it releases things like catecholamines from the sympathetic nervous system. And one of the effects that has is the activation of macrophages and macrophages and other innate immune actors as well. So microglial cells, for example, that function in the brain. So the activation of these immune cells will increase the expression of pro-inflammatory cytokines, will increase the expression of chemokines, which are chemical attractants of other inflammatory-based innate immune responders. It will increase the expression of adhesion molecules and increase the expression of acute phase reactants. Now, all of this will drive the expression of NF-kappa B in the brain. And when you have NF-kappa B in the brain expressed, you're going to reduce monoamine function, which is part of the reduction of stress, but you are going to increase the excitotoxicity function in the brain as well. So it keeps the brain at this sympathetic fight or flight state, right? So that's the basic premise of how external stressors impact the body. It does so through the HPA activation. HPA activation releases cortisol. Cortisol starts to create the stress response physiologically. At the same time, you're getting sympathetic activation. Sympathetic activation activates the immune system. And when the immune system is activated and increases inflammatory responses, it causes more inflammatory responses in the brain and increases excitotoxicity and reduces things like monoamine synthesis and monoamine function. So what does all of this mean? Well, the important thing to note here is what cortisol is at the end of the day supposed to do is as cortisol builds up, right? You have glucocorticoid receptors on all of the cells in the body, virtually all of the cells in the body, including all the cells in the brain. So as cortisol increases in the body and you get release of things like epinephrine and norepinephrine through the sympathetic activation, those things start binding all of the glucocorticoid receptors on the cells. They will start changing DNA expression to increase flight or fight response, but at some level of binding, that provides a negative feedback loop to the HPA axis to turn off, right? Because the presumption is that an external stressor is a life or death situation for a human, right? There's a tiger that all of a sudden jumps out of the bush. You need that external stressor to excite the body, to increase blood pressure, to increase heart rate, to run away from that stressor. But once you escape that stressor, you're supposed to be able to calm down from it, right? And that cortisol loop and the glucocorticoid receptor feedback inhibition or negative feedback cycle is the cycle that tells you to calm down and starts reducing the inflammation and actually starts reducing all of the sympathetic activation as well. So that's an important thing to note. Now, there's a big problem with chronic cortisol expression because one of the things that happens is when you get increased levels of cortisol, there is an increase in the permeability of the gut wall in leaky gut, which then facilitates more systemic inflammation, right? And this has been shown to be present in individuals with a dysbiotic gut. So if you have a dysbiotic gut, you have high levels of opportunistic pathogens, you have low levels of protective keystone strains. When you have higher levels of cortisol, it actually drives more dysbiosis in the gut and it drives more permeability in the gut. So now, like before, you would have LPS leaking through, which is causing all of the problems we talked about before. But on top of that, as your gut becomes more leaky, it's going to increase pro-inflammatory cytokines like interleukin-6, interferon gamma, interferon alpha, TNF alpha, and so on. When you have higher levels of these inflammatory cytokines, especially interleukin-6 by itself is known to come back and reignite the HPA axis. And over time, high levels of interleukin-6 will also downregulate the expression of glucocorticoid receptors, right? So now that really important negative feedback loop that glucocorticoid receptors provide, meaning as you have more glucocorticoids increasing in the blood, the receptors start binding them. The binding of the glucocorticoids by the receptors increases negative feedback, which tells the HPA axis to calm down. But the inflammation from the gut is actually reducing the expression of the glucocorticoid receptor, so you don't get that negative feedback. And on top of it, the inflammatory interleukin-6 is actually reactivating the HPA axis, right? So you lose the feedback mechanism and you increase HPA activation even more, right? So there's this massive hyperactive, hypersensitive HPA axis issue that goes on, which means that fewer, less intense stressors stress you out even more over time, right? And it takes less to activate the HPA axis, and it becomes harder and harder to come down from those stressors over time. So at some point in your life, you may have been relatively resistant to external stressors. Certain things, certain really important personal things may have stressed you out, but you may be able to work through it and calm down from it. But as time goes on, more and more things stress you out with a high amplitude, and it becomes harder and harder to come down from it, right? And that's part of what is being driven here through a dysfunctional gut, because a dysfunctional gut is not dealing with cortisol in the proper way. Evidence also suggests that these types of changes that occur cause a reduction in hippocampal serotonin levels, and as well as a reduction in BDNF expression. BDNF is brain-derived neurotropic factor, which is really important for regeneration and repair of brain tissue. As you guys know, serotonin is a happy hormone, all of which increases the susceptibility to depression, anxiety, and so on. So this continuous hyperactivation of the HPA axis, which is governed by a dysfunctional gut bacteria, because a dysfunctional gut amplifies the activation of the HPA axis from external stressors, right? A functional gut, a healthy gut with a very specific component to it, and I'll talk about what that component is next, but as a functional healthy gut with a very specific component can actually use cortisol to arrest the activation of the HPA axis and bring down the inflammation and actually bring down the physiological response as well. But a dysfunctional gut will amplify the impact of external stressors. So one of the ways in which a dysfunctional gut makes things even worse, right, is when you have simple issues like the metabolism of tryptophan in cells in the brain and other cells in the body as well. Tryptophan is a precursor to serotonin and melatonin, which is the happy hormone and the sleep hormone. And of course, serotonin, melatonin, and the other components that it can be metabolized to all tend to have neuroprotective function and trigger neuroprotective pathways. So they improve sleep, circadian rhythm, positive outlook, resilience to stress, good memory, and so on. That same starting material, tryptophan, that becomes neuroprotective through the right metabolic pathway can become neurotoxic if that metabolic pathway is shifted because of HPA activation, elevated levels of cortisol, and then inflammation from the gut microbiome and activation, overt activation of macrophages and microglia cells, right? So that inflammation from a dysfunctional microbiome and the continuous activation of HPA axis from the external stressors will actually flip tryptophan to this other pathway where it becomes kynurenine and then quinolinic acid, and that will actually be neurotoxic, where it increases depression, it increases irritability, it reduces resistance to stress, creates poor sleep and poor concentration as well. So simple biochemistry here showing that in one condition, when you have a healthy gut producing serotonin, melatonin, and differentiating tryptophan down those pathways, you can have the exact opposite when your gut is dysfunctional and you're getting constant triggering of the HPA axis because your gut is dysfunctional and you're not getting that feedback, negative feedback inhibition to reduce it. And that constant accumulation of cortisol in the body will trip the biochemical pathology towards this tryptophan being produced, it being differentiated into neurotoxic compounds instead. And we also know that LPS itself, if it's allowed to get in, will actually flip this pathology as well, that pathway. So LPS itself, which can activate an inflammatory response, can turn on that IDO pathway for the production of kynurenine from tryptophan, right? So having both LPS and chronic activation of the HPA axis is going to be a double whammy in ensuring that you're constantly producing neurotoxic compounds from what should be really healthy precursors like tryptophan. So then the question is, how do you address the stress-induced HPA activation and inflammation, right? Because it's hard to get away from stress. We can do things, and we should always do things to try to manage stress and the impact that stress has on us. But external stressors are always going to exist, and when they do exist, and when you experience them, they are going to activate your HPA axis, which is fine actually, because it's normal. It's a normal physiological response. The question is, can you come down from it? Right? Once it's activated, does the feedback inhibition work to reduce it so you can come down from it? Right? So that's a really critically important part. And as it turns out, the microbiome has a critical component and a very critical role in ensuring that we stop the continuous activation of the HPA axis and the propagation of all the inflammatory markers that result from that HPA axis activation. There is a very important compound made by certain bacteria within your gut called peptidoglycans, right? Peptidoglycans are basically a sugar, amino acid type of molecule that bacteria produce. And some of these bacteria produce it as an outer coating or like a little receptor that sticks out of the bacteria. And as it turns out, even in utero, peptidoglycans are so important for the development of the brain and the central nervous system. In fact, to a point where mom's gut bacteria will produce peptidoglycans that will end up in circulation, in mom's circulation. There are receptors for bacterial peptidoglycan on the placenta of the baby in utero that will bind these peptidoglycans, and the sensing molecules that will sense the presence of it, and transporter molecules that will actually grab it across the placenta and take it to the baby, and in fact take it to the baby's brain. And in the brain, there are receptors for these peptidoglycans. And when the baby's brain starts binding up the peptidoglycans, it stimulates neuronal development, right? It stimulates a proliferation of central nervous system cells, the differentiation into the different parts of the central nervous system, the spinal cord, the enteric nervous system, and so on. It increases synaptogenesis, which is creating more branches and more synaptic regions in the brain. And we know increased synaptic regions means increased cognitive function, increased intelligence and cognition and all of that, increased myelination, which is really important for nerves, to have that insulation, that myelin sheets that insulate nerve cells. And then it also forms the blood-brain barrier, right? So that's the important lining that helps protect the brain. Those are all things, critical, absolutely critical things that are being stimulated in the baby from bacteria from mom's gut, right? Think about it. The baby has peptidoglycan sensing molecules that the baby's DNA codes for and mom's DNA codes for. And the baby has peptidoglycan transporters that mom and baby's DNA codes for. And these are to transport a bacterial byproduct from mom's gut, which is one of the most critical products to ensuring the brain development of your baby, right? We have outsourced the brain development of the human baby in utero to a microbial byproduct. So just think about the importance and connection there, which is really, really fascinating, right? So if the mom doesn't have enough peptidoglycan producing bacteria, it increases the risk for neurological development issues and psychiatric disorders as well, right? So peptidoglycan is so critically important. And there's not a whole lot of bacteria that make this peptidoglycan layer. And so the idea is that, the peptidoglycan, if it's so important early on, and you have transporters and receptors for it early on as you're developing, maybe it also exists later on in life and it plays a role. And sure enough, it does. So there's a number of studies that have been done to show that peptidoglycans can reduce a number of inflammatory pathologies that are pathologies that impact things like IBS, chronic fatigue syndrome, depression, sleep, and so on, right? That's the interferon alpha, interleukin-6 activation, inflammatory activation that becomes a risk factor in all of these conditions. There's a number of studies that have shown that the presence of peptidoglycan, especially from Bifidobacterium longum, has the impact of reducing the inflammatory cycling of these conditions, right? So that is really exciting to see. And Bifidobacterium longum, not all of them contain peptidoglycan. It's a very, very unique strain that can actually make this peptidoglycan. And looking at an electron micrograph, this is what a normal Bifidobacter looks like that does not have the exopolysaccharide, which is made up of peptidoglycan. And then this is what a Bifidobacterium looks like when it does have the exopolysaccharide that is made up of peptidoglycans, right? So what we started to do is, once we've identified and isolated this organism, which was actually isolated from healthy normal individuals, comparing them to age-matched cohort that had mood disorders and so on, we find that people that do not have mood disorders, through the research institute that was looking at this, they ended up having more presence of this type of bacteria with peptidoglycan. So what we started looking at is in preclinical mouse models, and I'm not going to go through all of the studies, there's numerous studies on this topic, but in the interest of time, I'll just go through the summaries of the studies. In the preclinical animal models, 4 different published studies on this showed that the 1714 strain, that's the strain, it's Bifidolongum 1714. It's a unique exopolysaccharide containing strain that makes that peptidoglycan, which is the exopolysaccharide. It was able to induce interleukin 10 cytokine, which is the anti-inflammatory cytokine, which then helps systemically bring down inflammatory responses. It reduced a pro-inflammatory transcription factor, NF-kappa B, and even when the mice were injected with LPS to mimic that endotoxemia. So when the mice were given high levels of LPS, the strain was actually even able to reduce the inflammatory response to the LPS. It also reduced the inflammatory cytokines IL-1 beta, IL-12, TNF alpha in serum and in spleen as well. And again, even when challenged with LPS, which is one of the potent ways of inducing this. And it also reduced other pro-inflammatory cytokines when these mice were fed a high fat or obesity model diet, right? So in general, the conclusions from all of the mouse studies is that the 1714 strain is an anti-inflammatory strain with the potential to help with inflammation-mediated stress through HPA activation, because it's the same markers that are driving the chronic HPA activation. And then looking at the human clinical studies, there's a couple of different studies that have been completed and published. And what was shown was that the 1714 strain was able to reduce perceived stress and cortisol levels as well. There was a significant reduction in circulating serum cortisol levels. And then this was done in healthy male volunteers and was associated with a reduction in the pro-inflammatory tone of the subjects as well, which is demonstrated through the decrease of these key pro-inflammatory cytokines, IL-1 beta, IL-6, TNF alpha, and so on. And it does this through toll-like receptor stimulated whole blood from these subjects, right? So looking at these subjects, their ability to reduce the impact of perceived stress and reduce their the perceived stress from stressors, and then also reduce cortisol levels to improve their ability to manage stress, and then reducing that HPA activation as well. And then other brain activity studies has been done as well. This is a double-blind placebo-controlled study looking at brainwave activity under social stress conditions, right? So under social stress, people will go into a beta or delta dominant brainwave. What we want to be in is that theta wave, which is that flow state, really calm, high cognitive function. And when you add in the 1714 strains to individuals, it actually induces the theta wave function in these individuals when they're on the probiotic, right? And that's when they're undergoing a stress, an external stressor, in this case, a social stress. So what that showed was an enhancement of vitality, which is a given claim in certain parts of the world, and reduced levels of mental fatigue, because now the brain is in that theta wave mode, right? And so think about that. There is a microbe that, because of its production of exopolysaccharide and peptidoglycans, can arrest the inflammatory signaling in the gut, which is the inflammatory signaling that continues to loop and amplify HPA activation. And by doing that, it actually not only is reducing stress, it's reducing cortisol, but it's also changing brainwave function, right? It's taking people out of the beta wave band and delta wave bands and putting them more into the theta wave band, which is that calm flow state, high cognitive function. And what they showed is that activity of the increased activity of brain coping centers to counter-regulate negative emotions in stress. So this makes it a really important way in dealing with negative emotions or external stressors, that there's activity in parts of the brain that are supposed to counteract the responses to those. Those components of the brain were activated when you take the 1714 strain. So 1714 reduced perceived stress and cortisol output. We talked about that. It activates the centers of the brain correlated with improved vitality and control of negative emotions. It improves different indices of sleep, because there was also sleep studies, 2 different sleep studies done. So it improves the time to sleep, meaning it shortens the time to sleep and improves sleep quality in general. It also improves qualities of life parameters, such as improved social function and vitality as well. So it's making a significant impact and change on that HPA axis and the activation of the HPA axis. So the brain is profoundly affected by inflammation, which is likely the primary driver of most of the cognitive dysfunctions, and major causes of inflammation in the brain is a dysbiotic gut. We know that through LPS activation. We know that through continuous HPA activation and the amplification of the HPA activation through the gut, a dysbiotic gut. And then, of course, the resulting glucocorticoid receptor down regulation, which then makes it actually more easier to get stressed out, makes it easier for the HPA axis to get triggered and more inflammation to be released because of that. This presents to us a very unique opportunity to support the brain, because when you look at those two major pathologies that impact the brain in almost every way that's important to us in terms of our brain impact, it gives us an opportunity here to support much healthier responses in both of those pathologies, right? So we have to look at the stress-induced HPA activation and the LPS endotoxemia. We have to address both of those things to provide people with really comprehensive gut-brain support. And at the end of the day, we've got the ability to do that through different probiotic strains, because probiotics with the right type of probiotics have been shown to be able to do that. So in the case of the HPA activation, we have the new ZenBiome products, the ZenBiome Cope, which is the daily stress product, and then the ZenBiome Sleep, which also enhances sleep activity. And these can be used in combination if somebody is dealing with both daily stress and sleep, or you can just use the sleep if it's only sleep that's a concern, or just use the cope if only stress is a concern, right? And of course, you've got the Megaspore Biotic to deal with the LPS component of the endotoxemia. So between these two, we really are providing total cognitive support in individuals, because we're addressing all of the main physiologies and pathophysiologies that impact the brain in a negative way through the gut. Right. There's other things you might do as well. Obviously, mindfulness work and reducing stresses and removing yourself from stressful situations like relationships or workplaces and so on are all going to be really important. Diet and all that are going to be really important. But these organisms in particular have a unique ability to impact the two major sources of gut-brain dysfunction. Right, that's a really important tool to be able to have. And I can tell you, for those of you that have not seen the product yet, this is one of the most exciting products that we've launched, and it's showing up in the numbers. And in fact, we're getting feedback from our practitioners already, from individuals that have been on it from just the last 2 or 3 weeks. So it's really quite exciting. And I think the team is offering, for those of you that spent your time to come join us this evening, or who will be watching this recording over the next day or so, I think we're offering this very special thing. And I think, Morgan, maybe you can tell them a little bit more about this.
Yes, of course. So we have a 48-hour flash sale. So this is going to be 15% off your entire cart with the code masterclass 15. This is only for 48 hours. So happy shopping. This is really exciting that we're able to offer this to everyone who was able to tune in and be a part of this amazing presentation. Thank you so much, Kiran. You have a ton of questions that I would love to run through. Again, if we don't get through all of them, we will be able to answer them after as well, and then we'll try to get through as many as we can. So wonderful. All right. So one of the biggest questions that we got was kind of like, what is the protocol, right, for ZenBiome Cope and then ZenBiome Sleep, and then parting that with the Megaspore Biotic?
Yeah, so the protocol is really quite straightforward. So, if you have a new patient that hasn't been on either, and you've got the patient, they have mood issues throughout the day, they have issues dealing with stressors throughout the day, but they're sleeping perfectly fine, then what you do is just start them on their Megaspore, dealing, of course, with the leakiness in the gut, the LPS, and so on. And you would taper up the Megaspore like you normally would. And then you would start them right away on the ZenBiome Cope at 2 caps a day, taken at the same time. And in both cases, you could take them with food, right? So that's perfectly fine. Now, let's say that individual also on top of that had sleep issues, then what you would do is you would start them on the Megaspore like normal, but you would have them during the day take 1 ZenBiome Cope, and then in the evening before bed, take 1 ZenBiome Sleep. Right. So what that does is that ZenBiome Cope bottle is a 60 cap bottle. So in that patient, that Cope will actually last 2 months rather than just 1 month. And then the sleep product will actually last 30 days because there's only 30 caps in it. Now, if they come in and they have leakiness in the gut, and you have all the other gut issues that you want to deal with in LPS, and they don't have daily kind of mood disorders and stress, but they do have a hard time with sleeping, then you could just put them on ZenBiome Sleep. right? So you don't necessarily have to have the cope during the day, but you just put them on sleep. Most people we see require both, because if you have daily stressors and issues with mood throughout the day, it's also going to make it really hard to sleep at night. So for most people, they get one of each, and then they take one during the day and one at night, and then of course your Megaspore like normal.
Perfect. Perfect. And then now that you mentioned that, is there a time frame that people would be able to feel that impact, feel that effect? I know the clinical studies were done in 4 weeks, but just from your perspective.
Mm-hmm. Yeah. And so just taking empirical evidence, right? So we've had the product out officially to our healthcare practitioners. It's been about 3 weeks now. But prior to that, we've had it out to our employees for a month or so before that. So just from that empirical evidence, because as Morgan mentioned, the clinical trials all looked at 4 weeks impact, but most of the clinical trials were done around 2 billion CFUs. We have 5 billion CFUs per cap in the product because there is evidence, we have other evidence that there is a dose response. So the higher the dose up to a certain point, the faster and the more impactful the response. So for our practitioners, we wanted to go with what we would consider to be the doctor grade of the product, which is 5 billion CFUs per cap. So that's measurably higher than what was done in the clinical trials. So from that, what we're seeing is that most people are reporting a noticeable impact in the first 7 to 14 days. Especially with the sleep product, in the first week, people are noticing an impact. And on the cope product, within the first 14 days or so, people are noticing quite a bit of an impact. And some of the unique things, and some of this actually comes from our employees to a certain degree because they've been on it longer. Things that you might notice is just like more vivid dreams at night, so you're going into REM sleep more, you're having better quality sleep, more efficient sleep. And then waking up during the morning, people have to do less to get their day started. So less coffee in the morning, less time kind of sitting around in bed, and trying to get moving, right? Because that lethargy, that lack of energy is all part of this pathophysiology. So don't be surprised if your patients report back to you within 6-7 days that they're feeling different, and that's been super exciting for us.
Yeah, that is very exciting, especially for this huge new novel probiotic. I mean, that's huge. Would children and mothers that are pregnant, are they able to take this as well?
Yeah, so think about mothers who are pregnant, right? They need that peptidoglycan. That peptidoglycan is so important for the neuronal development of the child. So it would be absolutely applicable to moms taking it, and moms can take it at the same dose that any normal adult would. Children can take it as well, and in fact, I would recommend maybe 1 capsule a day for kids, or maybe you can even pull apart the capsule and quickly mix it in some food and take it in if they want to go with half a capsule a day. I think even that would be quite impactful on kids. So it's absolutely applicable to pregnant women and kids.
Perfect. And then someone asked, do you work directly on brain inflammation while working on the gut microbiome and leaky gut? You were talking about how it's going to all coincide each other. So if we're combating the leaky gut, we're going to combat the leaky brain, right?
That's exactly right. So the vast majority of brain inflammation comes from the gut, right? So whether it's through the LPS mediated pathway or the HPA activation of macrophages and microglia cells, all of that centers around the gut as a checkpoint for brain inflammation. If it wasn't for the gut, outside of like direct neurotoxic things like chemicals from the outside, the brain would really have very little inflammation, right? And we know that the brain has its own lymphatic system, it has its own immune cells, but all of those come to the gut and get activated before they go back to the brain. So as you're taking care of inflammation in the gut, you are automatically taking care of inflammation in the brain.
Perfect. And then are you able to talk about the other ingredients in the probiotic itself? Because I know we're having the saffron extract, we have the lemon balm, we have the L-theanine, we have a ton of different great extracts in there as well.
Yeah, so we put a few things in there that would just help some of that immediate symptomology, right? So for people, so the lemon balm and the saffron are really great with helping reduce some of the more immediate symptomology in people that are excited, that have sympathetic activation. The theanine really does help with sleep, and it becomes supportive to dealing with the core root problem, right? So at the end of the day, the big thing that we have to address is that activation from the gut. And so we're addressing that with the 1714 strain, but at the same time, we wanted to enhance people's feeling and perception relatively quickly so that they actually feel better. And those are ingredients will actually help enhance those more immediate feelings. But again, those things actually don't work as well unless you're dealing with the core problem. So we're finding that combination of dealing with the core problem of the gut, which drives the dysfunction, actually will make the lemon balm and the saffron and the theanine work better, because they can do their job without the gut combating their positive functions.
Right. And then the Bifidobacterium lagom is a live probiotic, correct?
It is, yeah, and it's unique in when it comes to Bifidobacterium because it has that exopolysaccharide that's made up of peptidoglycan, and that provides it a coating and extra stability and protection so that it can actually survive through the gastric system and then get to the site of action alive. As you can see, there's a little bit of a theme with microbes that have special coatings that have unique functionality, right? Our whole spore-based products are based on microbes that have that unique coating. So it's not the same as a spore coating, but it has its own version of a unique coating.
Perfect. And then one of my favorite articles that you did mention was the mice with the short bouts of stress. So even if somebody may not feel stressed all day long or have that chronic stress, even those short bouts of stress, this would be able to kind of benefit and support somebody with that.
That's right. Yeah. Because short bouts of stress all will activate the HPA axis, right? And if their gut is dysfunctional, each time they experience a short bouts of stress, it can actually continue to amplify the inflammatory response from that short bout of stress for longer and longer periods of time. Because remember, if your gut's not healthy, it becomes very hard to come down, not only from the physiological effect of that stress, but the inflammatory component of that stress as well, right? So continuous short bouts of stress with the dysfunctional gut will actually perpetuate more dysfunction in the gut, will create an opportunity for pathogenic organisms to start expressing virulence factors, which all of that will continue to activate your HPA axis even more. So each of those short bouts of stress actually become longer and longer to recover from.
And that could even lead to like sleep problems, right? I mean, this is all going to even circle even more.
That's right. I mean, sleep is really a secondary effect of daily stress, because when your sympathetic system is activated, which is what happens when your HPA axis gets activated, your sympathetic system works against the parasympathetic system, which is the rest and digest system, right? Because you have that sympathetic parasympathetic toggling, that tone. And so HPA activation from a dysfunctional gut, from interleukin-6, from that chronic low-grade inflammation is going to continuously keep your sympathetic system turned on, which means it becomes really hard to go to the parasympathetic and utilize things like serotonin, melatonin to calm down, GABA to calm down, and to actually go to rest. So dysfunction in sleep is really a prolonged effect of chronic stress.
Perfect. And then do the new products affect mucin development and repair?
It can in a positive way. We haven't done a study specifically on mucin repair. However, when you look at the ability of the new products to reduce those critical inflammatory cytokines, especially things like TNF alpha, NF kappa B, interleukin-6, and then the ability to increase IL-10, those are all the types of changes that are good for rebuilding the mucin layer. So even though we haven't done a study specifically on what do the new products do to the mucin, we know from that mechanism of action, it's going to be beneficial to the mucin layer. So it pairs greatly with the TGR kit, right? So if you have, and you've been using the Total Gut Restoration Kit with your patients, you add this in, it adds that next layer of resolution to those pathways that affect that patient. And in this case, it's going to be most profoundly noticed in their mood and their sleep.
Perfect. I have another one. Is the Zenbiome cope beneficial for individuals that have eating disorders that suffer with multiple mood challenges?
Yeah, that's a great question. So we have not studied it in eating disorders specifically, right? However, there's lots of studies that show that the inflammatory pathology that leads to mood disorders is the same inflammatory pathology that's present in anorexia nervosa and other eating disorders. So I would assume, that it's going to help in some degree, but I can't say definitively that we have a study that showed this, right? But the pathology is the same. Anorexia nervosa, major depressive disorders, anxiety, all of them are driven by the same type of pathology.
Perfect. And then what is the best way to test for LPS in serum?
Yeah, unfortunately, you can't do a direct LPS test commercially. LPS is a real finicky molecule. Once you pull it out of serum, you pull the serum out of the body, it degrades pretty quickly within about 4 hours. You have to freeze the sample at minus 80 degrees in order to be able to test the LPS later on. So that's not practical in a clinical setting. So you can look at LPS surrogate markers. So the best known surrogate markers for LPS are LBP, LPS binding protein, and there are labs that do immunological assays for LBP. You can also look at certain immunological markers like soluble CD14, interleukin 6, and interleukin 1 beta. So if your patient tends to have elevation in chronic elevation in serum LPS levels, they will also have elevation in soluble CD14, interleukin 1 beta, and likely interleukin 6 as well.
Wonderful. And then kind of going along with that, what do you recommend is the best test for gut permeability?
Here's the interesting thing. I mean, LPS endotoxemia is the gold standard of demonstrating permeability, right? Showing that you're getting translocation of LPS from the lumen, past the intestinal lining into circulation. That's the gold standard. That's what studies the NIH and American Diabetic Association, all of the biggest studies on this topic of intestinal permeability look at LPS translocation. Since you can't really do that really practically in the clinic, my recommendation would be to assume that the vast majority of your patients have intestinal permeability, right? Because they likely do. When we did our first leaky gut study, we were looking at healthy young individuals. So these are individuals that were of normal body weight, no conditions that they're being managed for. They weren't on any medication, so they're healthy, normal individuals. 55% of them had really severe leaky gut, right? So if 55% of those people have really severe leaky gut, the types of clients and patients you guys see, almost all of them will have severe leaky gut, right? So it's just so prevalent nowadays that I don't think you even have to bother testing for it. I think you can presume they have it, and there's nothing wrong with approaching therapies that we know improve leaky gut, like increasing diversity, increasing keystone species, short chain fatty acid production, and all that. Those will all be the things that improve the leaky gut, but they also do a number of other physiological beneficial things in the body.
Perfect. All right. Well, if we did not get to your question, that is okay. We will be able to go through them and answer them through email as well. And then I just wanted to thank Kiran so much for doing this amazing presentation. It was incredible. And then everyone, please remember to use the code masterclass 15 for 15% off your entire cart for the next 48 hours. And then this is also a first of a new series, so be on the lookout for the next masterclass coming soon. And then actually another plug for mini keynotes is coming up September 30th from 3 to 6 Eastern time. So this is going to be a free virtual event. You get free CEs with that as well. So the registration link for mini keynotes will go out with this email as well when you get the recording in the upcoming dates. But other than that, this was fabulous. Thank you so much.
Thank you so much, everybody. We really appreciate you joining us today. And then, as Morgan said, we'll answer the questions that didn't get answered and we'll make sure we get it out to you. And if you're watching the recording and you come up with questions from the recording, send it to us. I think info@Microbiome Labs. All of those questions will come to Morgan and myself. We'll make sure to answer it for you. Thank you, Morgan, for hosting us.
Thank you so much. Thank you so much.
And running the program. I appreciate it.
Thank you. Have a great evening. Bye, everyone.
Bye.
Thank you.